Yeah, I needed to hurry you along, David, because I'm the next speaker. So, so my name is Andy Marks. I'm chair of Physiology and Cellular Biophysics, and I'm going to tell you about our work on Alzheimer's-like signaling in the brains of COVID-19 patients. I am going to be talking about a drug that we call a reichelt, and so I have a conflict of interest to disclose because we have a company, Armgo, that's developing these drugs. So I'm going to really switch gears for everybody and introduce you to this calcium channel here called the rhinitine receptor. It's an intracellular calcium release channel found in most cell types, and it's on the SR or ER. It's the largest known ion channel, and it releases calcium required for muscle contraction, neurotransmission, and many other bodily functions. And the yellow molecule is a stabilizing subunit called calstabin, and the green one is calmodulin. And this is a high resolution structure that we solved using cryo-EM recently. Okay. The rhinitine receptor is shown here. It sits on the sarcoplasmic or endoplasmic reticulum. And in skeletal and cardiac muscle, it's activated when the muscle cell is electrically stimulated. It releases calcium required for muscle contraction. And this is the process of excitation, contraction, coupling. But it also is present in brain. And over the last 20 years or so, we have shown what occurs when there's a stress-induced leak in the calcium release channel in different organs. And this is associated with a loss of the stabilizing subunit calstabin. When the leak occurs in the brain, it can promote seizures and cognitive dysfunction in the heart failure and arrhythmias, in skeletal muscle, muscle weakness and muscular dystrophy, cancer-associated muscle weakness, and it can be associated with diabetes and pancreatic beta cells. So with this very quick background, we wanted to explore the potential role of leaky rhinitine receptors in Alzheimer's disease, because there have been a long story in the literature about calcium abnormalities in Alzheimer's. And what I'm showing you here is biochemistry that we call the biochemical signature of leaky rhinitine receptors, in this case, the RYR2 isoform in human Alzheimer's disease brains. And the biochemistry that I'll be showing you was done by Stephen Reichelt in the laboratory. And what we're looking at here is an immunoprecipitation of the rhinitine receptor from the brains of the Alzheimer's patients. And we're showing that in the Alzheimer's patients, there is phosphorylation, pKa phosphorylation, nitrosylation, oxidation of the channel, a reduction in another subunit of the channel called spinophyllin, which is a targeting protein for the phosphatase PP1, and a decrease in the stabilizing subunit calstabin. And this again is what we call the biochemical signature of leaky rhinitine receptors based on many, many studies in the laboratory. We also were interested in the effects of the reichelt drug that I mentioned. And here shown in the upper left are two forms of reichelt, S107 and ARM210. And using cryogenic electron microscopy, Zephyr Melville in the laboratory has recently identified the binding site for the reichels. It's out here in the periphery of the channel in a cleft where there's also a molecule of ATP bound. And here is shown the binding of the reichelt drug. And through an allosteric long distance mechanism, this stabilizes the closed state of the channel and prevents that pathologic leak of calcium. So we then went to look in two murine models of Alzheimer's disease. These are models of familial Alzheimer's disease. One is an APP-PS1 and the other is a triple transgenic model. And again, what I'm showing here is biochemistry indicating the biochemical signature of leaky rhinitine receptors with pKa phosphorylation, nitrosylation, oxidation of the channel, a reduction in the stabilizing subunit calstabin. And when we treated the animals with the reichelt S107, we could restore the binding of calstabin. And we know from many studies in the laboratory that, that this fixes the leak in the channel. And this occurred in both murine models of Alzheimer's disease. We could also fix the leak by crossing the mice with a knock-in of alanine residue into the PKA site at serine 2808. So a single amino acid substitution in the rhinitin receptor prevented the leak in the channel. And so there's both genetic rescue and pharmacologic rescue. When we looked at the leak of calcium out of microsomes from the brains of these murine models of Alzheimer's disease, there was an increased leak of calcium. This is an assay where you make microsomes, you put a fluorescent calcium indicator outside, and you block the uptake of calcium after loading the microsomes with ATP. You block the pump, and there's a passive outward leak of calcium. And this was increased in the Alzheimer's mice, but after you treated with the S107 or did the genetic cross, we could normalize the leak or prevent the leak. On the lower panel C, there's single channel recordings where under conditions where wild type channels are tightly closed with a low open probability of 0.01, the Alzheimer's channels had, or channels from the Alzheimer's mice had about a 10-fold increase in open probability consistent with a leak of calcium. And again, that could be prevented by treating the mice with the S107 drug. We then looked at the cognitive and behavioral dysfunction that characterizes these models of Alzheimer's disease. And in the fear conditioning, there's an auditory stimulus which is given, and then the mice, after 24 hours, are placed back into the same context. And the wild type mice will freeze when they hear the same sound, and the Alzheimer's mice either don't learn or don't remember, and so they don't show any fear conditioning. And we could restore the normal responses to the fear conditioning, again, with either pharmacologic or genetic rescue. Similarly, in a Morris water maze where the mice are tasked with finding a hidden platform, the Alzheimer's mice either don't learn or don't remember where the platform is, and the treatment either with a pharmacologic or genetic rescue restored their ability to find the hidden platform. Novel object recognition is where you have a mouse exposed to two objects, and then after a period of time, put them back into that environment with a new object, and the normal mice will discriminate between them and will preferentially explore the new object. The Alzheimer's mice, again, either don't learn or don't remember that, and you can restore that to normal with treatment with a drug or a genetic rescue. In an open field test, the same type of thing occurred where normal mice spend more time in the center of the field, and treatment restored that normal behavior. And finally, in an elevated plus model, wild type mice spend most of the time in a closed arm as opposed to an open arm, and this abnormal behavior in the Alzheimer's mice, where they spend more time in an open arm, which is considered less safe, and that could be fixed with either genetic or pharmacologic rescue. We also looked at the electrophysiologic manifestations of Alzheimer's disease with both long-term potentiation and long-term depression. Long-term potentiation can be thought of as memory storage, and long-term depression can be thought of as memory editing, both of which are abnormal in Alzheimer's. And again, these abnormalities were corrected either by pharmacologic treatment of the mice to fix the leak in the channel, or by crossing them with mice that don't have a leaky rhinitin receptor. And finally, we looked at amyloid deposition in these mouse models. In the APP PS1 mouse model, they express it for a human form of amyloid, which can be detected with an antibody, and that's shown here in panel B. And again, either treatment of the mice with S107 or crossing them with a non-leaky rhinitin receptor mouse prevented the deposition of the amyloid, as shown here and quantified in the rest of the slide. And then we generated a mouse with a constitutive, mimicking a constitutive PK phosphorylation of the RYR2 channel, which induces a leak. So these mice are born with leaky channels. And they also had the biochemical signature of leaky channels. And they had the same abnormalities that I just described to you in the Alzheimer's mice. So abnormal fear conditioning, performance on the water maze, novel object recognition in the elevated plus maze, and even defective LTP and LTD rather. And so what this shows is that merely by introducing a leak into the rhinitin receptor calcium release channel in the brain, you can reproduce many of the features of Alzheimer's disease in these mice. So taken together, this introduction by way of background makes the case that if you have a leaky rhinitin receptor in the neurons in your brain, this calcium leak can be upstream of pathways that lead both to neurofibrillary tangles and amyloid deposition by activating these calcium activated enzymes, CAMKK2, calpain, and so on. And this can create a vicious cycle where you develop a leak in the rhinitin receptor, which can be toxic to neighboring mitochondria, inducing them to produce more reactive oxygen species, which further oxidize the rhinitin receptor and exacerbate the leak. And upstream of this can be activation of beta-2 adrenergic receptors, which have been linked to amyloid deposition, resulting in phosphorylation of the channel, promoting the leak. And this can be inhibited with the drug, reichelt drug S107, which prevents the loss of the stabilizing subunit, fixes the leak in the channel, and restores normal behavior, cognitive function in these murine models. So with that by background, we then wanted to look at patients who had died from COVID-19 because of the emerging story of brain fog and long COVID. So there are a lot of limitations to this study. So first of all, these are patients who did not have long COVID by definition, since they died from the COVID-19. And only 3 of them were under the age of 60. And as you can see from the pathology, many or most of them had severe hypoxic brain damage. But nevertheless, we went on and we wanted to study these human brains from the COVID patients. And what we found initially was evidence of increased oxidative stress and inflammatory and adrenergic signaling in the brains of the COVID-19 patients. I should mention that we looked, as others have, for the evidence of virus in the brain samples, and we did not see any. But there was evidence of increased inflammation and oxidation. So for example, the GSSG to GSH ratios were increased in cortex and cerebellum. Kynuritic acid, marker for inflammation, increased as well in cortex and cerebellum. There was activation of the phosphosmad signaling pathway in the COVID-19 patient brains, as shown here. And CAM kinase 2 activity and PK activity were also increased in these COVID-19 patients. So a phosphorylation of tau has been widely studied as a contributor to neurodegeneration. There are multiple phosphorylation sites on tau, and as shown in this cartoon, they can contribute to multiple changes that have been linked to neurodegeneration. So we looked at evidence for phospho tau in the brains of the COVID-19 patients. And indeed, if you look down here in panel C and the quantification over here in D, there were multiple sites on tau that were phosphorylated. In this experiments, we tried to look at old versus young patients, so the cutoff being 60. And you can see that in some cases, the tau phosphorylation was increased in cortex and cerebellum in both the quote unquote older and younger patients. And in other instances, it was only increased in the older patients. We also looked at signaling related to amyloid production. And we did not find any abnormalities there in terms of the precursor, APP, precursor to amyloid, or the base enzyme involved in amyloid. So these studies suggested that phospho tau could be increased in the brains of COVID-19 patients, but we didn't see evidence for amyloid. We then looked again at the rhinitin receptor shown here in the brains of the COVID-19 patients, and found it to be oxidized, phosphorylated, nitrosylated. And we could also prevent that by treating the brain tissue ex vivo with another reichelt drug called ARM210, as shown here. We then looked at the channel function, and in this instance, we used ryanidine binding, which is a surrogate marker for activation of the channel, since ryanidine binds preferentially to active channels. And we showed that under conditions where the wild type or channels from control patients were not active, that the channels from the COVID-19 brains had increased activity, which could be reduced again by treating the brain tissue with ARM210. So this would be indicative of leaky channels. There was also a reduction in calbindin, which is a calcium binding protein, and increase in GCP2, which could promote the production of PKA. So reduced calcium binding could potentially exacerbate the effects of a leaky calcium release channel, and the GCP2 elevation could promote PKA phosphorylation of the ryanidine receptor. So in summary, what we're proposing is that the inflammasome linked to SARS-CoV-2 infection, which has been widely reported by many labs, can result in activation of the TGF-beta phosphosmad pathway, oxidation of the ryanidine receptor, and also phosphorylation. This can create a leak in the ryanidine receptor, which can create a vicious cycle where there's mitochondrial calcium overload, ROS production, further oxidation of the channel. And our preliminary data with a very small number of patients suggests the possibility that this could promote abnormal phosphorylation of tau, which could be playing a role in the cognitive dysfunction observed in patients suffering from long COVID. We also found evidence of leaky ryanidine receptors, by the way, in the hearts and lungs of these same patients, suggesting that there could be a role for this calcium leak in the observed abnormalities in cardiovascular function and potentially also in the lung defects. So I want to observe that there are a lot of caveats to the study, a tiny number of patients. Most of them were older, evidence of hypoxic brain damage in most of them. And obviously a lot more work needs to be done before confirming this, but the suggestion that Alzheimer's type signaling could be playing a role in long COVID, and that this could be linked to leaky calcium channels. I want to acknowledge the terrific work of the brain autopsy team at Columbia University, shown here. Obviously a huge effort to characterize the brains from these COVID-19 patients. Draw your attention to this important article recently published by this group, in which they looked more extensively at the neuropathology in the brains of the COVID-19 patients studied at Columbia. And Jim Goldman and Peter Knoll were particularly helpful to us in the study. The folks in my lab are shown here, and I mentioned the bulk of the biochemistry done by Stephen Reichen in the lab, single channel studies by Chi Wan, structural studies by Zephyr Belleville and Marco Mioto. And again, I want to acknowledge the important contributions of the pathology department and Peter and Jim, the autopsy staff, the COVID bank at Columbia, and support from these entities. So I will stop there and be glad to take questions.
Super, Andy, you're well under time. You didn't need to worry.
Okay, try to catch us back up.
Yeah, all good.
Yep.
I'm curious about the extent to which it's workable to fiddle with something as central and as widely important as the receptor,
Mhm.
in terms of side effects. I wonder
Sure.
where we are with the drugs, in terms of that.
So great question. So the drugs preferentially bind to leaky channels and have very little activity on the normal channels. So for example, the Bmax is increased almost tenfold when you oxidize and phosphorylate the channels. And there's one form now that's in clinical testing, just completed a preliminary study, phase 1B, it's called, at the NIH, without any safety warnings, and that was in patients who have mutations in the skeletal form of the channel and cause muscle weakness and a progressive form of myopathy. We have another study planned in patients with mutations that make the heart channel leaky and have exercise-induced sudden death. So in terms of safety profile, it seems to be well tolerated in phase 1 studies. And also in animal models, when we give huge amounts of the drug, no effect on heart rate, blood pressure, things like that. So it seems that the drug is essentially restoring the normal function of the channel by preventing the leak without blocking or activating any abnormal pathways.
So that's a high bar for the drug, and I guess that's amazing it passes them.
Yeah, no, it's very lucky.
Hey, Andy, if I could ask a couple of
Yeah.
questions.
Sure.
In your original mouse model, can you comment quickly? I'm sure you said this and I just missed it. What causes the reduced binding of calstabin to the receptor in the first place?
So it's induced by stress, and that's manifested as post-translational modifications of the channel that are typically phosphorylation and oxidation. The exact mechanism of that, we're not completely sure, but preliminary structural data suggests that the PK phosphorylation shifts the channel into what we call a prime state, which is halfway between closed and open. So it renders the channel more susceptible to activation, and this could explain the leak. And we're currently exploring whether or not the binding affinity of the calstabin to that prime state could be reduced. So that's our current hypothesis.
And if I can ask a couple more,
Sure.
Can you comment very quickly on how you guys discovered reichelt?
Yeah, so there was a compound called JTV519 that had been made by Japan Tobacco and abandoned, and it got picked up by a group of scientists at Yamaguchi University who had read one of our papers and were testing a number of compounds to see what could fix the leak in the rhinitin receptor. And so in their survey, they identified JTV519, which was not attractive for clinical development because it has a lot of off-target activity. And so we developed compounds that were based on that originally, and then sort of branched out into a chemical library that currently has about 3,000 compounds with varying activities, but very little off-target activity for the ones that we're developing.
And are there animal models for COVID infection that exhibit neurological defects?
Yeah, a very tough problem because, by definition, most of the animal models are set up to do studies like we heard from Dave Schultz, to look for drug. And so they are, short-term models by definition, looking for the ability to prevent infection and so on. So there really aren't good models for long COVID that we're aware of. We've also looked for models for heart, also difficult to study. So that's one of the challenges in this field.
And if I could ask one last one.
Sure.
In terms of reversibility, I'm assuming in the animal model, if you take a stressed animal and then let them relax for a while, everything goes back to normal. Is that true?
Yeah, so most of these stress models, are chronic diseases like heart failure, diabetes, cancer, so If you induce stress artificially, so we did a study on post-traumatic stress disorder where you put the mice in chronic restraint, and there you can reverse the effects of stress if you take them away from the stress signal. So
Yeah, I guess what I'm driving at is, to what extent do we anticipate the neurological effects of COVID, especially in older patients, to be reversible?
yeah, I mean, no idea, but in the mouse models, we were able to both prevent amyloid deposition, for example, and reverse it in those, mouse models of familial Alzheimer's disease, but really have no way to predict what would happen in patients, older patients.